Welcome to this video on biology mock revision for the 2025-2026 academic year. We will be covering essential topics, including cell biology, microscopy, eukaryotic and prokaryotic cells, magnification calculations, and practical microscope skills. Let's focus on mastering these details for your upcoming exam. This slide provides a visual overview of the core biological concepts we need to review, ranging from cell structures and DNA to metabolic processes and physiological systems. Use this as a map for your revision. Let's start with a foundational question. What are the similarities and differences between eukaryotic and prokaryotic cells? Key similarities include the presence of a cell membrane, DNA, and ribosomes. Notable differences involve organelles like the nucleus, mitochondria, and chloroplasts, which are typically absent in prokaryotes. Understanding magnification is crucial. The equation is magnification equals image size divided by actual size. Remember your units. 1 millimeter equals 1,000 micrometers, which equals 1 million picometers. How does an electron microscope differ from a light microscope? Electron microscopes offer significantly greater resolution and magnification, allowing you to see fine details like mitochondria. However, they cannot image living cells. They produce non-colored images, and they can even generate 3D representations. When using a microscope, you can improve image clarity by staining the sample with solutions like iodine or methyl blue. Once the slide is prepared, use the coarse focus knob for initial adjustment, followed by the fine focus knob to sharpen the image. To describe the structure of DNA, highlight these key features. It has a double helix shape, a sugar-phosphate backbone, and complementary base pairs where adenine pairs with thymine, and cytosine pairs with guanine. What is the lock and key mechanism for enzymes? It is a model where each enzyme, acting as the lock, is specific to a particular substrate, which acts as the key, ensuring precise biological reactions. This graph illustrates enzyme activity relative to temperature. Initially, activity increases as enzymes gain kinetic energy. After reaching an optimum temperature, the activity drops sharply because the enzymes begin to denature, causing their active sites to change shape so the substrate no longer fits. Here are the equations for aerobic respiration. The word equation is glucose plus oxygen produces carbon dioxide and water, plus 38 ATP. The symbol equation is C6 hydrogen oxide plus 6 O2 yields 6 CO2 plus 6 H2O, also with 38 ATP. For anaerobic respiration, the equations differ by organism. In animals, glucose is converted to lactic acid with the release of 2 ATP. In plants, glucose is converted to ethanol and carbon dioxide. Why do muscle cells contain more mitochondria? Because these cells perform significant work. They require more energy. Mitochondria are the sites of aerobic respiration, the very process that produces the ATP needed for that energy. In digestion, specific enzymes handle different nutrients. Amylase breaks down carbohydrates and starch into glucose. Protease breaks down proteins into amino acids. And lipase breaks down lipids into fatty acids and glycerol. Photosynthesis is vital for plant energy. The word equation is carbon dioxide plus water, in the presence of light, produces glucose and oxygen. The symbol equation is 6 CO2 plus 6 H2O yields C6 hydrogen oxide plus 6 O2. Finally, remember the 3 limiting factors of photosynthesis: carbon dioxide concentration, light intensity, and temperature. Each can restrict the rate of the process under different conditions. Moving on to transport mechanisms, red blood cells facilitate gas exchange through diffusion, where particles move from high to low concentration. They are specifically adapted with a biconcave shape, abundant hemoglobin for oxygen transport, and a lack of a nucleus to maximize space. The rate of diffusion is influenced by 3 key factors. Higher temperatures increase particle kinetic energy, leading to faster movement. A larger concentration gradient speeds up the net movement, and a greater surface area provides more space for particles to pass through. When a potato is placed in pure water, osmosis occurs. Water molecules move from an area of higher water potential in the solution into the lower water potential within the potato, causing the potato to gain mass. To calculate the percentage change in mass, subtract the original mass from the final mass, divide that result by the original mass, and then multiply by 100. In this example, 2 divided by 7 times 100 gives us a 29% increase. Looking at this graph, the concentration of sucrose within the potato can be identified where the change in mass is 0. Following the data, we find this point is 0.25 moles per cubic decimeter. Root hair cells are specialized for efficient absorption. Their long, hair-like projections significantly increase the surface area available for the uptake of water and minerals through osmosis, diffusion, and active transport. These images highlight the structural diversity of cells involved in plant transport and absorption, illustrating how each type is uniquely shaped to support its specific function within the plant. We can distinguish between 3 types of blood vessels based on their adaptations. Arteries have thick walls with elastic fibers to withstand high pressure. Veins have large lumens and valves to ensure one-way flow. Capillaries are extremely thin, only 1 cell thick, to allow for efficient gas exchange. A hole in the heart can cause fatigue because it allows oxygenated and deoxygenated blood to mix. This reduces the amount of oxygen reaching the body's cells, leading to decreased aerobic respiration and lower ATP production. This diagram of the heart shows the key structures, including the chambers, valves, and the major vessels: the aorta, pulmonary artery, pulmonary vein, and the vena cava. Each plays a critical role in circulating blood efficiently. Transpiration is the movement of water through a plant. It begins with water entering the roots via osmosis, traveling upwards through the xylem vessels, and finally exiting through the stomata in the leaves as water vapor. The xylem and phloem are specialized tissues. Xylem cells are dead, providing rigid structural support and a clear path for water transport. Phloem cells are living and use energy from companion cells to actively transport sugars, with sieve plates acting as selective barriers. Mitosis is essential for growth and repair. It involves duplicating the genetic information, breaking down the membrane, lining up and pulling apart the chromosomes using spindle fibers, and finally dividing the cell into 2 identical daughter cells. A reflex action occurs in a rapid sequence. A stimulus is detected by receptors, triggering an impulse in a sensory neuron, passing through a relay neuron in the spinal cord, and traveling via a motor neuron to an effector which performs the response. Blood glucose is controlled by insulin and glucagon. Insulin instructs the liver to store glucose as glycogen, lowering blood levels. Conversely, glucagon triggers the liver to release stored glycogen as glucose, raising levels. This is a classic example of negative feedback. Let's review the roles of key hormones. Adrenaline triggers the, quote, fight or flight response, instructs the liver to release stored glucose, and increases your heart rate. Thyroxine, on the other hand, is primarily responsible for controlling your basal metabolic rate. The menstrual cycle is regulated by 4 key hormones. FSH matures the egg, estrogen builds up the uterine lining, LH triggers the release of the egg, and progesterone maintains the lining throughout the cycle. Hormonal contraceptives work by utilizing synthetic progesterone. By keeping progesterone levels high, the body's natural cycle is suppressed, which prevents the release of an egg, effectively preventing pregnancy. These images represent various biological systems and cellular structures we've covered, providing a visual summary of the complexities within physiological and transport processes. This collection of imagery illustrates further examples of biological structures and experimental setups, helping to reinforce the concepts of cell specialization and scientific inquiry. Mastering scientific vocabulary is essential for your exams. Remember the difference between independent, and control variables, how to address random errors, and why precision and accuracy are critical when evaluating your practical results. We have covered a significant amount of material today. Take some time to review these topics, practice your calculations, and stay confident. Good luck with your revision and your upcoming exams.